Okay, now we have developed that notochordal process. Here I can show you a transverse or axial view of the embryonic disc. Here's up where the amnion is, here's down where the umbilical vesicle is, and here's the ectoderm, the mesoderm, and endoderm, and here we have that notochordal process. And so the notochord is important because it is going to induce the formation of the neural tube that happens on the dorsal side of the embryo. So it's going to actually communicate and induce these cells, communicate with and induce these cells that are overlying the notochordal process to actually make these cells turn into the neural tube, and that's going to develop into the brain and the spinal cord. So that's why the notochordal process is important. And so we're going to continue the development of the neural tube and the brain and the spinal cord in another session. I just wanted to give you sort of a picture of where we were going with this information. Okay, so mesoderm is going to further differentiate into regions that are going to specifically develop into certain structures. And I want to give you the heads up on that as a whole. And I talked to you about mesoderm being intraembryonic, and so on this figure here you can see that we have here's where the notochord is, and here we have mesoderm. They just colored it so because it's going to develop into certain structures, and then there's mesoderm here, intraembryonic mesoderm there, and there's intraembryonic mesoderm here. And you can see that it's going to shift to yellow, and this is not intraembryonic mesoderm, it's actually extraembryonic mesoderm that's sandwiched between the two layers that are going to be lining the amniotic cavity and the umbilical vesicle. And so it's not going to be within the embryo. And so you can see that the transition when you're looking at this view, this is a dorsal view looking downwards on the developing embryo, is really not super dramatic. And so it's continuous with extraembryonic mesoderm, and I wanted to point that out. So when we look at the region of mesoderm that is adjacent to the notochord, we see that highlighted in yellow here, and that's called paraaxial mesoderm. And paraaxial mesoderm is going to very specifically develop into structures called somites, and there'll be an entire session on somites and what they develop into. And so they are, just as a big picture ticket item here, they're going to give rise to the axial skeleton, so not your appendages, but your trunk, your axial skeleton, and all the associated musculature, and the dermis of the skin, of all the skin. And so these somites are going to be a big, or play a big role in embryonic development for all of your axial skeleton and all of your dermis of the skin. And so what that looks like here, if you look at this section, that is a frontal view of the embryo. Here you can see that these are the somites developing here, and the first one is going to develop just a little bit caudal to where the otic placode, or the section that's going to develop into the ear, is located at, and they're going to keep proliferating in a caudal region here. And you can see how this paraaxial mesoderm that's going to develop into somites is nearby where the neural tube is going to develop. So any neurons that need to go into this block of musculature is really kind of next door, so they can grow out into the developing somites because it's in very close proximity. And this is what it looks like on an actual embryo. Here you can see the developing neural tube. You know, we talk about these systems in isolation, but really everything's developing at the same time. We're just going to come back later and visit the neural tube development. And here are the developing somites along the dorsal aspect of the embryo that are actually developing from paraaxial mesoderm. And so you can see how the somites proliferate and actually increase in number all the way up until the end, where there's an approximate number of 42. So we're going to continue the development discussion on somites. All right, the intermediate mesoderm, which is the next one when you're going from medial to lateral, the next block of mesoderm, it's a little bit smaller, is going to give rise to the urogenital tract. And that's all you have to remember. Intermediate mesoderm, urogenital tract. When I learned embryonic development, the professor who gave the lectures on the urogenital tract, his name was Dr. Swartz, and so we called the intermediate mesoderm of Swartz, so that no matter what, you could always remember what intermediate mesoderm turned into, because he was the one who lectured on that material. It did kind of help, I have to say. There were a few times I really needed to have something to hang my hat on when it came to intermediate mesoderm. So you can relate it to any kind of anything that has to do with the urogenital tract, just so that you can hold on to the fact that this block of mesoderm will turn into those systems. And we'll pick that up later on in the course. And then we have lateral mesoderm. Lateral mesoderm is the most lateral intraembryonic mesoderm, and here you can see in this image how it's continuous with extraembryonic mesoderm here, and that's going to be cut off eventually when we have folding of the embryo, but at this point you can see that it's still continuous. And it's going to, this lateral mesoderm is going to further divide into a block that is going to be more dorsal, and the block that's going to be more ventral. And you can see that these spaces that start to form, coelomic spaces, coalesce with each other and become a almost cavity here that we are going to call an intraembryonic coelom. And so don't worry, we're going to show you this again. All right, and so the development of the coelom doesn't only happen in this area where they have lateral mesoderm. It also happens up in the cranial aspect of the embryo in the midline. You can see this entire area where the coelom occurs. And this is the region that's called the cardiogenic mesoderm, because it's going to develop into the heart and associated structures. So all of this coelom that develops and expands, it kind of forms a horseshoe shape that we refer to as the intraembryonic coelom. And this is important for forming our body cavities. And so here you can see if this embryo was cut down the mid-sagittal cut and we were looking at it from either this way or this way, here you can see the little tiny wee part that you would see being the intraembryonic coelom, because all of this part is continuous. And then if we were to section the embryo here at the area where the coelom communicates on the sides, you can actually see this section where it communicates right here. So it's a lot bigger of a space, and it's communicating freely with the extraembryonic coelom. So we have this space inside that is a horseshoe shape, goes all the way up through the midline, comes on the other side, and communicates outside of the developing embryo with the extraembryonic coelom that was talked about in the first 2 weeks of development. All right, so this just is I like to give you a big picture. Where am I going with these instead of having something to hold your hat on? That this coelom is going to develop into the pericardial cavities and the pleural cavities and the peritoneal cavities. So this is how we end up having body cavities. And you'll have a whole lecture or session that's going to revolve around the development of these cavities. So right now it's just important to think about how they develop, and it's also really important to think about the fact that they are divided into two sections of lateral mesoderm. And the reason why is because of innervation. Because this lateral mesoderm is separated by the coelom into two layers, it's not just regional where these layers end up being that's important. It's that the actual type of innervation that is attached to the tissue that develops in these two regions are functionally different. And so when we have separated the mesoderm into mesoderm that is adjacent to the ectoderm, so this is more superficial mesoderm, we're going to call that mesoderm somatic mesoderm, and we're going to call the mesoderm that's adjacent to the endoderm here the mesoderm that's adjacent, we're going to call that splanchnic mesoderm. And these terms are going to start to become more and more familiar to you as you continue your understanding of the nervous system. So the somatic mesoderm with the overlying ectoderm, that's going to form the embryonic body wall. So the outside of the body, the wall, that makes up the perimeter or periphery of the body. And we're going to refer to those tissues together as the somatopleural, and that's going to have functionally GSA, or general somatic afferent innervation. The splanchnic mesoderm that is next to the endoderm is together with the endoderm referred to as the splanchnopleural, and the endoderm here is going to develop into the gut. And so with that mesoderm that is adjacent to it, we're going to call, this is going to be functionally innervated by GVAs. And so these functional differences are going to continue as you understand the nervous system in its multiple layers. And it all sort of started right here with the division of the lateral mesoderm into the body wall stuff and the gut stuff, and inside viscera that's going to be derived from the gut tube. It's more than just the gut. And so somatopleural is going to eventually go in a GSA functional component region or direction, and the splanchnopleural is going to be associated with GVAs. All right, and so I want to do one more thing before I ask you a couple questions. I want to briefly discuss with you a structure that you'll see in figures that's called the allantois. The allantois is a rudimentary structure in human development, but you do see it in figures here, and it has somewhat of an important purpose, but they don't really explain very much of its function in the textbook, so I just wanted to give it its own little stab here so it's not confusing to you because it's never really discussed. So it's a small diverticulum that happens in the umbilical vesicle, and it's going to extend outwards into the connecting stalk. So later on, this connecting stalk will eventually develop into the umbilical cord. And so this diverticulum just extends for a while. And it's going to remain relatively small and doesn't really have a whole lot of function in humans. But it is important in regards to the fact that it has some surrounding mesoderm around the allantois, so in this region. And it is going to develop, this mesoderm will develop into the umbilical arteries and will also be one of the first sites that's going to produce blood cells. And so I did want to talk about, even though the allantois doesn't really have a lot of function, it will become a remnant structure in the adult human called the urachus. So you'll learn about it later, what it becomes. But it doesn't really have a huge function, but the mesoderm around it does, because it will develop into the umbilical arteries and also does make blood cells. So there it is, the allantois.